Majority of flowering plants use a range of animals as pollinating agents. Bees, butterflies, flies, beetles, wasps, ants, moths, birds, sunbirds and hummingbirds and bats are the common pollinating agents. Figure 2.11b. Among the animal, insects, particularly bees, are the dominant biotic pollinating agents. Even larger animals such as some primates, lemurs, arboreal, tree-dwelling rodents, or even reptiles, gecko lizards, and garden lizards have also been reported as pollinators in some species. Often, flowers of animal pollinated plants are specially adapted for a particular species of animal. Majority of insect pollinated flowers are large, colorful, fragrant, and rich in nectar. When the flowers are small, a number of flowers are clustered into an inflorescence to make them conspicuous. Animals are attracted to flower by color and or fragrance. The flower pollinated by flies and beetles secrete fall odor to attract these animals. To sustain animal visits, the flowers have to provide rewards to the animal. Nectar and pollen grains are the usual floral rewards. For harvesting the rewards from the flower, the animal visitor comes in contact with the anthers and the stigma. The body of the animal get a coating of pollen grains which are generally sticky in animal pollinated flowers. When the animal carrying pollen on its body come in contact with the stigma, it brings about pollination. In some species, floral rewards are in providing safe places to lay eggs. An example is that of the tallest flower of Amorphophilus. The flower itself is about 6 feet in height. A similar relationship exists between a species of moth and the plant yucca, where both species, moth and the plant, cannot complete their life cycle without each other. The moth deposits its egg in the locule of the ovary and the flower in turn gets pollinated by the moth. The larvae of the moth come out of the egg as the seed start developing. Outbreeding devices. Majority of flowering plants produce hermaphrodite flowers and pollen grains are likely to come in contact with the stigma of the same flower. Continued self-pollination results in inbreeding depression. Flowering plants have developed many devices to discourage self-pollination and to encourage cross-pollination. In some species, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma become receptive or stigma become receptive much before the release of pollen. In some other species, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both these devices prevent autogamy. The third device to prevent inbreeding is self-incompatibility. There is a genetic mechanism and prevents self-pollen from the same flower or other flower of the same plant from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil. Another device to prevent self-pollination is the production of unisexual flowers. If both male and female flowers are present on the same plant, such as castor and maize monoecious, it prevents autogamy but not geitonogamy. In several species, such as papaya, male and female flowers are present on different plants, such as this plant is either male or female dioic. This condition prevents both autogamy and geitonogamy. Pollen pistil interaction. Pollination does not guarantee the transfer of the right type of pollen. Compatible pollen of the same species of the stigma. Often, pollen of the wrong type, either from other species or from the same plant, it is self-incompatible, also land on the stigma. The pistil has the ability to recognize the pollen, whether it is of right type, compatible, or of the wrong type, incompatible. If it is of right type, the pistil accepts the pollen and promote post-pollination events that leads to fertilization. If the pollen is of wrong type, the pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube growth in the style. The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of continuous dialogue between pollen grain and the pistil. This dialogue is mediated by chemical component of the pollen interacting with those of the pistil. It is only in recent years that botanists have been able to identify some of the pollen and pistil components and the interaction leading to the recognition, followed by acceptance or rejection. As mentioned earlier, following compatible pollination, the pollen grain germinates on the stigma to produce a pollen tube through one of the germ pore. The content of the pollen grain move into the pollen tube. Pollen tube grows through the tissue of the stigma and style and reaches the ovary. You would recall that In some plants, pollen grain are shared at two-celled condition, a vegetative cell and a generative cell. In such plants, the generative cell divides and forms the two male gamete during the growth of the pollen tube in the stigma. In plant which shade pollen in three-celled condition, pollen tube carry the two male gamete from the beginning. Pollen tube, after reaching the ovary, enter the ovule through the micropyle and then enter one of the synergid through the filiform apparatus. Many recent studies have shown that filiform apparatus present at the micropylar part of the synergid guides the entry of the pollen tube. All these events from pollen deposition on the stigma until pollen tube enter the ovule are together referred to as pollen-pistil interaction. As pointed out earlier, pollen-pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition of the pollen. The knowledge gained in this area would help the plant breeder in manipulating pollen-pistil interaction, even in incompatible pollination, to get desired hybrids. You can easily study pollen germination by dusting some pollen from flowers such as pea, chickpea, Crotalaria, balsam, vinsa on a glass slide containing a drop of sugar solution about 10%. After about 15 to 30 minutes, observe the slide under a low power lens of the microscope. You are likely to see the pollen tube coming out of the pollen grains. As you shall learn in the chapter on plant breeding, chapter 9, a breeder is interested in crossing different species and often genera to combine desirable characters to produce commercially superior varieties. Artificial hybridization as one of the major approaches of crop improvement program. In such crossing experiment, it is important to make sure that only the desired pollen grain are used for pollination and the stigma is protected from contamination from unwanted pollen. This is achieved by emasculation and bagging techniques. If the female plant bears bisexual flowers, removal of anther from the flower bud before the anther dehisces using a pair of forceps is necessary. This step is referred to as emasculation. Emasculated flowers have to be covered with a bag of suitable size, generally made up of butter pepper, to prevent contamination of its stigma with unwanted pollen. This process is called bagging. When the stigma of bagged flower attain receptivity, mature pollen grains collected from anther of the male parent are dusted on the stigma and the flower rebagged and the fruits allowed to develop. If the female parent produces unisexual flowers, there is no need for emasculation. The female flower buds are bagged before the flowers open. When the stigma becomes receptive, pollination is carried out using the desired pollen and the flower rebagged. Double fertilization. After entering one of the synergid, the pollen tube releases the two male gametes into the cytoplasm of the synergid. One of the male gamete moves toward the egg cell and fuses with its nucleus, thus completing the syngamy. This results in the formation of a diploid cell, the zygote. The other male gamete moves toward the two polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus, PEN, figure 2.13a. As this involves the fusion of three haploid nuclei, it termed as triple fusion. Since two types of fusion, syngamy and triple fusion, take place in the embryo sac, the phenomena is termed double fertilization, an event unique to flowering plants. The central cell after triple fusion becomes the primary endosperm cell, PEC, and develops into the endosperm, while the zygote develops into the embryo. Post-fertilization structures and events. Following double fertilization, events of endosperm and embryo development, maturation of ovule into seed and ovary into fruit are collectively termed post-fertilization events. Endosperm. Endosperm development precedes embryo development, while the primary endosperm cell divides repeatedly and forms a triploid endosperm tissue. The cells of this tissue are filled with reserve food material and are used for the nutrition of the developing embryo. In the most common type of endosperm development, the PEN undergoes successive nuclear divisions to give rise to free nuclei. This stage of endosperm development is called free nuclear endosperm. Subsequently, cellular formation occurs and the endosperm becomes cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water from the tender coconut that you are familiar with is nothing but free nuclear endosperm made up of thousands of nuclei. And the surrounding white kernel is the cellular endosperm. Endosperm may either be completely consumed by the developing embryo, example pea, groundnut, beans, before seed maturation, or it may persist in the mature seeds, example castor, coconut, and be used up during germination. Embryo. Embryo develop at the micropylar end of the embryo sac where the zygote is situated. Most zygote divide only after a certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo. Though the seeds differ greatly. The early stages of embryo development, embryogeny, are similar in both monocotyledons and dicotyledons. Figure